Welcome back to our CAPE Biology Past Paper Review. Today we are continuing with Unit 2, Paper 2 from the year 2019. For this review, we are going to review question number 2. We have already reviewed question number 1. Question number 2 is from Module 2, which is about biosystems maintenance, which is dealing with homeostatic properties. Question 2, part 1 reads that impaired glucose tolerance is a risk factor for certain diseases. An oral glucose tolerance test was administered to persons with normal glucose tolerance, which is represented as NGT, and impaired glucose tolerance, which is IGT1 and IGT2. Now, blood plasma glucose levels were monitored at 30-minute intervals for 2 hours. The data are given in Table 1. And so Table 1 represents plasma glucose levels following oral glucose intake for groups of persons with different glucose tolerance levels. NGT, which is normal glucose tolerance, and IGT1 and IGT2, which are impaired glucose tolerance. And so the chart here represents our data information. And part 1 of this question says, on the grid provided on page 11, plot a line graph for the data provided in Table 1 with time on the x-axis. And this is a 6-mark question. And so I have already done the graph to make it easier and faster. And so the graph here is illustrated. First, you must have a title to indicate what the graph is showing. So the graph is showing plasma glucose levels against time. Also, what is important, you need to label your axis. So very importantly, your y-axis will be your glucose levels in millimole per liter. And your x-axis will be time. And also a point to note here, which is kind of dull, but there's a key. And on my key, my x-axis is showing 4 centimeters, represent 30 millimoles per liter. And on the y-axis, which is 1 centimeter, is actually 1 millimoles per liter. And on the x-axis is 4 centimeters for 30 minutes. All right, so the time is on the x-axis, just to point it out. Now the graphs were plotted, and you notice our NGT, which is our normal glucose tolerance, and our impaired glucose tolerance levels, which is represented by IGT1 and IGT2. So you have the fine broken lines for IGT1 and the longer broken lines for IGT2. Again, just to point out, you must use fine points to join your coordinates, and also straight unbroken lines, or when I say unbroken, I should say double lines. The line's supposed to be smooth and not broken. So you use your ruler to make those proper drawings to make sure you have continuous lines that are fine lines. All right, again, to emphasize that your axes must be labeled properly and well identified. Now, let's go back to the questions. And for part 2 of this question, it states that 2 key observations, state 2 key observations that can be made based on the results of this study. And if you should look at the graph carefully, let me go back and forth with the graph. And if you look at the graph carefully, you notice that NGT is always below the IGTs. All right, so that is one thing you can observe based on the result. But before I get into the conclusion, based on the narrative of the question, I just want to point out a few things real quick. And so one thing I want to point out right here is about glucose levels, and then I'll get into the conclusion shortly. But what is meant by impaired glucose tolerance? This is where you have the glucose level in the blood above normal. So I'm going to say glucose above normal. And when I say glucose, I mean glucose in the blood. The glucose in the blood, let me just write that out to make sure that you understand. The glucose in the blood, right, is above normal, but the person does not have diabetes, okay? So I'm saying, but do not have diabetes, okay? So that's to make a note of that. It can also be presented in another way as well. You can also say it is between normal and diabetes, okay? So the glucose level is moving from between normal, you can say between normal and diabetes, okay? All right, so also you can also state it to be pre-diabetic. So this is also what they call pre-diabetes as well, okay? All right, so those are ways that you can describe the term impaired glucose tolerance. What I also want to point out to you before I jump into answering the first part of the question, which is the conclusion based on the data, is that talk about glucose levels. So let's talk about glucose levels a little bit. And so glucose levels that you need to be familiar with is and we're measuring this in terms of millimoles per liters. And so one of the first things that you must remember is that the values between 4 and 6 is pretty much what they call excellent level or very good level. So this would be excellent. Let's say excellent. That's really, really good. And then if you have between 7, say if you go between 7 and say about 8, then what you have here now, you can consider this as being good. So this will be good. And if you have here 9 to 14, then this will be poor. Now, anything below 8 or up to 8 is really considered a good level. So once you have those levels, you consider yourself as being in good conditions. Once you go in 9 and above, then you have some concerns. All right, so let's go into the conclusion part of this now to give our 2 conclusion based on what they asked us to do. It says, state 2 key observations that can be made based on the results. So our observation based on the graph, again, just to look quickly, notice it that the NGT is below and the highest point and the NGT here is 9. Okay, so a matter of fact, it's slightly below 9, which is really good. All right, and so the first conclusion that we can make right here is that normal glucose tolerance persons, I'm just going to say NGT for short. So NGT, so normal glucose tolerance level persons, right, always tend to be below 9, and this would be 9 millimoles per liter, okay? And this will be even 9, and let's say even, because based on the data chart, it was after oral intake, so even after oral intake of glucose, okay? All right, so great. Now, the other thing that we can conclude right here is that persons with IGT, which is impaired glucose tolerance, whether it be 1 or 2, their glucose levels, and just I'm going to go to the graph and show you this real quick. Their glucose levels will generally be above 9, will be above generally 9 millimoles per liters, okay, after oral intake of glucose, okay. And also what you can make in terms of a conclusion that NGT glucose levels will be lower than persons with IGT. So that's another observation that you can also make right here, that NGT for the same timing will actually be less than IGTs, okay? All right, just to make a note of that. All right, and also it is shown on the graph as well. The NGT is lower for the corresponding time compared to IGTs, okay? All right, so and again, if you notice the IGT points, they are generally tend to be above 9 if you notice it. And remember, after 9, you consider yourself to be pre-diabetic or diabetic, all right, or you have some concerns that may need to check out. All right, so now let's go to the next part of the question. And the next part of the question here, let's go down to that. All right. Now that's the chart there. So yes, it's explain the mechanism by which glucagon and insulin function to maintain normal concentration of glucose in the blood. Now, the first thing I want to tell you about glucagon and insulin, they are two hormones that are produced by the pancreas. However, they are produced by the islet of Langerhans. Okay, so I'm going to make a note of that. So both of them are produced. So both, all right, I could put this in, then put this in red. All right, so as a side note right here. So this is not even answering the question. I just want to give you this bit of information right here. So both are produced by the islets of Langerhans. All right. All right, by the longest of Langerhans in the pancreas, okay? All right, and so it's very important for us to note that. What I also want to point out, because they're also produced by specific parts within the pancreas as well. And so I'm going to make a note right here that glucagon, glucagon is produced, the glucagon is produced by what I call the alpha cells. So these are produced by the alpha cells, just to make a note there. And insulin is produced by the beta cells, okay? Just to make a note right there. All right, so now let's jump into the question now and start answering this question here. And again, we're looking at the mechanism, right, in which glucose and insulin function to maintain normal concentration of glucose in the blood. And so the very first point that I want to make right here is that they both work in what I call a negative feedback system. So the first point I'm going to make is that glucagon and insulin, okay, they work in a negative feedback system, okay, in a negative feedback system, all right, to control blood glucose, okay, or glucose in the blood. All right, what I want to point out right here, what I mean by negative feedback, is that as one of these hormones is increasing, the other one is decreasing. So in other words, they work oppositely to bring about the same action or function. So the action is that to control glucose. If glucagon is increasing, insulin is decreasing, and vice versa. Now the next point to make right here is that insulin, so this is our next point, is that insulin uh, is responsible to decrease, so this will decrease the glucose in the blood, right? The glucose in the blood. All right, so that's the purpose or the function of insulin, to decrease the amount of glucose present in the blood. While glucagon, let me put this here, while glucagon, all right, increases the amount or concentration, can use concentration as well, amount of glucose in the blood, okay? All right, so again, insulin is responsible to decrease it, while glucagon is responsible to increase it. All right. Now, let's be a little bit more specific now in terms of what is happening. And so the next point I'm going to make here now is that when glucose is high, so when, all right, we'll go back a little bit. Okay. All right, so when, come back. All right, so when glucose content, I'm going to say glucose content is high, right? When glucose content is high, insulin will be secreted or released. The insulin is released, right? And convert, we're going to convert now the glucose and convert glucose into what they call glycogen, okay? Into glycogen. All right, so that's what is very important right there. So long as glucose content is high, then the insulin will be responsible to convert glucose into glycogen. And just to make a note right here, because we're moving from a simpler substance to a more complex substance, this is an anabolic process or reaction, all right? Just to make a note right there as well. Now, also what is important while this is happening is that triglycerides, okay? So triglycerides, the triglycerides are produced, so they are produced as well. So triglycerides, they are also produced, okay, or synthesized in the liver, just to make a note of that, okay? So triglycerides are synthesized. All right, and the glucose. So once glucose convert to glycogen, you have triglycerides are being synthesized, and through all of this process here, then glucose will decrease. Okay, so once the glucose is being converted to glycogen, then glucose will decrease. Okay. All right. So the next point now, let's go to the next point now and talk about when glucose is low. Okay, so here now when glucose is low, all right, then the opposite, technically the opposite would happen. Okay, so here now glycogen will be converted into glucose, but let me first put that glucagon, okay, say glucagon, so you're going to say glucagon, all right, is secreted. All right, so glucagon is secreted, which will catalyze glycogen. So it's catalyzed glycogen. All right, let me say catabolized let me put it right here, catabolized glycogen, and this will be into glucose, just to make a note of that. So catabolize glycogen into glucose. All right, so when this happens now, which is very important, so you catabolize it into glucose. And what is important here as well is that fatty acids, okay, just to make a note right here, that fatty acids, all right, are released as well, okay? Fatty acids are released. And once fatty acids are released, they will inhibit the release of glucagon, okay? All right. So the release that inhibits glucagon. All right, and so that's a very important process as is taking place right there. That the fatty acid will inhibit the glucagon. All right, so that's very important to note. All right, and so as a result of this, then you have an increase in glucose. Okay, so here you have an increase in glucose right here. Okay. All right. So that answered that. Now let's go to the next part of the question now. And the last part of this question, I think it should be yes. It said, comment on the clinical significance of the presence of glucose in the urine. So what benefit in terms of information, or how can they use the presence of glucose in the urine to make predictions medically? Is that the presence of glucose indicates that a person may be diabetic. All right, not say the person is necessarily diabetic, but may be diabetic. So the presence of glucose indicates that the person may be diabetic. All right, we're not saying the person is diabetic, but may be diabetic. All right, or I could just say here, or need some medical assistance or need other tests. Okay, so there may be need for the test or need to be tested. Okay, need to be tested for, and I'm gonna say for like renal disorders, because the person can have damaged infections and so on, or in the kidneys could be damaged as well. Different aspect of the kidneys could also be damaged. All right, and so, or maybe malfunctioning. So I'm gonna also say that the person is able to make reference to if they need to be checked, so if there's any renal failure or renal damages and so on. All right, so that will answer that part of the question right there. So it's very important for a person to understand the content of glucose in the urine, so therefore they could get the necessary treatment early enough and to prevent further damages and so on. All right, so it's a very significant thing in terms of detecting. If glucose is not detected, then you can confirm that the person have a proper working kidneys, and definitely the person is not diabetic. All right, so it serves real good function in terms of helping to predict what is happening to a patient. All right, so let's go to the next part of the question now. And this is for part B. It said control mechanism involving regulation of regulation in plants. It said these control mechanisms have commercial applications with reference to the properties of ethylene. Analyze its action to bring about desirable changes in fruits during commercial ripening. So what I want to point out here, because this is key, we're looking with reference to the properties of ethylene, and based upon the properties of ethylene, how can it bring about desirable changes in fruits during the commercial ripening process? All right, and so I think it's just fair for me to outline a few properties of ethylene so we can actually use them to guide our response in terms of how it will enable desirable changes during the ripening processes. All right, and so the first thing I want to talk about in terms of properties, I want to make reference that ethylene is a gaseous substance, right? In fact, I could also say that it's the simplest alkene gas, right? Because only 2 carbons, right? Just to make a mention of that, and it is from the alkene homologous group. Also, what I want to mention here in terms of properties is that ethylene will have low melting point, so they have low melting point here, right? So low melting point, and the low melting point, let's go over a little bit more. The low melting point is really about negative 169. Okay, that's a very low melting point there. And this is in degrees Celsius. So I'm going to put this, let's say degrees Celsius there. All right, just to be right in mind. All right, so degrees Celsius. And also another point that I could make right here as well is that it has low boiling point as well. Not just low melting point, but also low boiling point, okay? All right, so low boiling point. And the boiling point here is negative 104 degrees Celsius as well, okay? Just to make a note of that. And the last point I want to make in terms of properties here, that it has a low mass, a relatively low mass. And again, because it's the simplest alkene, then yes, it has a relatively low mass. But I'm going to put low mass for short right now. All right, so the concept now of how these things enables ethylene to bring about desirable changes during ripening, being a gas and also lightweight. So I'm put that one as 1 because gas tend to be lightweight anyhow. So being gaseous, but I'm going to talk about some specific changes that actually take place in the ripening process at the end of it. So it's being gaseous and lightweight. It really serves a very important function right here. And again, it is the simplest alkene. I could put this in bracket for you as well. So it's the simplest alkene. Okay. All right. So it's the simplest alkene right here. All right. Simplest alkene. Okay. All right. Now, also what I want to do in terms of this is I'm going to refer to the function of this. And so here we can talk about being the simplest alkene or being a gaseous substance, right? It spreads really easily, okay? So just to make a note of that. So it spreads easily. And because of this spreading, all right, if it spread evenly, then therefore it can also easily, therefore it can cover foods evenly. Okay, so spread easily and cover, so let me go all the way out to the end and cover the fruits evenly. All right. So they end up moving very fast and end up covering the fruits that you want to ripe evenly to bring about here now uniform ripening. And this is very important, you know, because if you think about it, if it was a slow moving gas or a heavy gas, then it will be difficult for it to move and spread over a large distance, and hence the fruits may ripe with different colors, right? So one part may be right before the other part, but since it can move so fast in small spaces and covering all the fruits, then yes, it may cause uniform ripening in the fruits. Also, what is important here again is that due to low melting point, let's think about the melting point, right? So let's put an asterisk, right? So due to low melting point. All right, and let us put both of them together because this substance will be a gas at those temperatures, really low temperatures, right? So due to low melting and boiling points, okay, so let's put both of them together, and boiling points, all right, it will remain a gas, it will remain a gas, all right, under normal conditions. So on the normal temperature and pressure, it will remain a gas, okay, under normal conditions. But I must say under normal conditions, and you can say under normal temperature and pressure, right? All right, STP. All right, so being a gas under normal conditions, therefore that this gas will be less likely to be condensed. I must say no condensation. Not saying that is impossible to condense, but under normal conditions, you have no condensation of this gas. And since you have no condensation of this gas, that means the particles of ethylene, okay, the particles of ethylene will move rapidly, right? Will move rapidly at higher temperatures or standard temperatures, okay? So they tend to move rapidly. And remember, this is also based upon the kinetic theory of matter as well, that as temperature increases, the particles will move faster due to more kinetic energy, and so they move more rapidly, all right? And so therefore, if they move rapidly, they can spread, okay? They can spread over a larger surface area, a larger area, okay? Over a larger area. And the thing about commercial will have commercial food ripening will be a lot of fruits. So therefore, spread over a large area in a short time. And so therefore, this will make it be effective to ripe a large amount of fruits in a very short time. So that desirable change could be a large amount of fruits, could be get ripe or ripen in a short period of time. Okay, so in a short time. All right, so that's a very important thing. So instead of you have a whole storage of fruits that you want to ripe within a week, instead of having them ripen, some don't ripen, all that, you could have a whole batch of fruits being ripe at the same time, and so therefore those could go on the shelves looking uniformly colorful and nice. So the first point talk about the uniformity in ripening. The second part is talking about the large amount being ripened at the same time, okay? And so some desirable changes that we can actually make reference to in terms of the properties of ethylene as well. So I'm going to make a note of these. And these changes may include brighter colors. So I'm going to make a point there so you can have brighter colors. Okay, so you have brighter colors. So instead of having, like, let's say the fruit's supposed to be yellow or orange. Instead of having part green and part orange or part yellow, the entire fruit could have a uniform, nice, brilliant, vibrant color. Okay, so a next thing that we can have here in terms of desirable changes is that you can have complex sugar, or you can say complex carbohydrates or complex sugars convert to simple sugar, okay, to simple sugars. All right, and so I want to explain this a little bit because, like, for example, some fruits before they are ripe, they tend to be more starchy, all right, and also sour. But when they're ripe, what happened to them is that they become more sweet. And because remember, you can't taste complex carbohydrates, only simple carbohydrates that actually taste sweet. So the complex carbohydrates, they're unsweet, okay, but the simple ones, they are sweet. All right, and so for this reason, riper fruits tend to be sweeter than unripe fruits, okay? So the complex sugars can be converted into simple sugars that will make the fruits actually taste better, sweeter, okay? So what will happen here, because of the ethylene, it will induce respiration, it will induce the conversion of sugar, all right, and also metabolic processes that will enable sugar to be formed. Also, what is important as well is because of the ethylene, it can also stimulate the production of aromatic compounds. So aromatic compounds are formed, and this is very important because this will make fruits have a better smell too, because the ripe fruits, you get that nice smell from the fruits, and so that will also create more attraction towards the fruits as well. All right, instead of having the sour, unpleasant, dull colors, they'll be bright, sweeter, and better smelling. Okay. All right, so those would be the points that I will put there in terms of answering this question. All right, so let's go into now the last part of this question right here. And the last part of this question, which is part C, is that the control of the water content, which is the water potential of the body, is called osmoregulation. It's in the space below, construct an annotated flow chart to illustrate the mechanism by which the body responds to reduce water content, which is low water potential. Your flow chart should identify the effector, receptor, regulator, and response. And so what I'm going to do here is to just quickly draw the flow chart first, and then what I'll do is put some arrows to indicate what is going next. So I'm going to start from a point that is not totally necessary because they actually tell that there is low water potential already, but I want to start from where it begins. And so the first thing that may happen here to trigger this response, and notice I'm using the word trigger this response, is that you have an intake of salt, or you may have excess sweating. Okay, so you could have excess sweating as well. I will also indicate different points. We have different things. And so the next thing that you can have here, you can have because of excess sweating or a large intake of salt, what you're going to have here is low blood water content, okay, or low water in the blood. Okay, so low water in the blood. All right, and so the next thing that you may have after you have low water content in the blood is that a significant trigger, and again, I'm using the word here to kind of indicate what is going to happen here. So you're going to have a trigger of something by the brain, and the brain is a part of the nervous system that is able to control or regulate what is happening. I'm just kind of using the word to kind of bring out the points which I'm going to add after this. And so what you're going to have here is the pituitary gland. Okay, all right. The pituitary gland will release what I call ADH. So the pituitary gland will secrete or release. Okay, you can use the word secrete, which is nice, but I'm going to say release or secrete, don't matter really. So the pituitary gland releases a surge of ADH, okay, of ADH. And ADH here is what I called anti-diuretic hormone. So this is anti-diuretic hormone, okay? All right, and so very important for us to note that. And so once the brain releases this trigger, to control, because now this is now the control part of it, or the regulation part of it. So here we're going to release ADH, and ADH now will now cause a response. So I'm using the words as I go along. Will cause a response by now causing a high volume of water to be reabsorbed. So high volume of water is being reabsorbed by the kidneys. Okay, so the kidneys will reabsorb these water or high content of water. Very important, right? And then the next thing that I want to make note of right here, because the kidneys now are reabsorbing high content of water, the next thing that will happen here is that only a small amount of urine will be released, and this is an effect now. So an effect of it now will be small amount or small volume, small amount or volume of urine is released in the bladder, okay? Is released in the bladder. All right, so very important to know that. And I must say at this point that if there's small volume of urine is released in the bladder, this urine that is released is also highly concentrated because the water is reabsorbed by the kidneys. So here the urine is really concentrated. So I'm going to say concentrated urine right here. Okay. All right. It's not going to be a much of urine, but it's going to be concentrated. Okay. So concentrated urine. Okay. All right. So great. Now the next thing will happen here. We're not finished yet. We're almost there though. The next thing that will happen here after that is that you have a high volume of water released in the blood from the kidneys now. Okay, because remember the kidneys reabsorb the water, so therefore you'll have a high amount or volume, okay, or volume of water is released into the blood. Okay. All right, so great. And there's one more point that I'm going to make right here. The last point, and is that here now you'll have normal water content in the blood. So this will be now normal water content in the blood. Okay. All right, so that's exactly what happened there. And so just let me quickly put some arrows in, and as I put my arrows in, I might just indicate quickly what is happening here in terms of here. Let me put this right here. Let me put this in red. What you're going to have here in terms of once you intake the salt and excess sweating start taking place, then the nerves will pick up these changes like excess salt, excess sweating, and so here you're going to have we have receptor, all right, being detecting these changes. Okay, so these changes will be detected by receptors. And then now, as I mentioned to you earlier, is that the brain now, in terms of the pituitary gland, this is where you have the regulation taking place. Okay, so the brain will regulate what will happen here. So this is a regulator. Let's say regulator right here. All right, and so as a result, then there's a response by the kidney in terms of absorbing or reabsorbing water. So this will be a response right here. And then what will happen here now is that in terms of a muscle action by the tubules of the kidney and also the bladder, is that these will be what they call effect by movement of so this will be the effect they're going to cause right here. And so let's call this effector. Okay. So this is where we're going to take place because the muscles again in the bladder will respond to the condition of releasing only a small amount of urine. All right. And also high volume of water content again will be the change that takes place and result in normal water content in the blood. All right, so folks, I hope this shed some light into the question. But again, as I say, flow diagram, what you need to put is your arrows to show you what first, second, and third. So I'm going to put them in real quick. And then we're out of here. I really wish you luck in your examination. And please just try to do well. And just try to remember all that you have been doing right through the academic here, regardless of the difficulties and the circumstances that some of you may face. I do sympathize. But again, do well. Catch you up another time. All right, stay safe and stay blessed.